Thank you so much, Alex, for the introductions, and thank you for being here today. So, I want to talk to you a little bit about something I've been working a little bit during my postdoc, and I've slightly changed the title as tissue flows and matrix interactions during morphogenesis. So, the reason I titled it this way is um, I've generally been interested in flows across multiple scales. And what I mean by flows are flows which could be just proteins which flow within an organism or an in vitro system. So here are two examples. One, on top, you have flows between different proteins in an in vitro reconstituted system, whereas you could have similar sort of flows of proteins inside a developing C. elegans oocyte at the same time. And similarly, you could have these tissue flows which emerge in a cell culture context in response to mechanical cues. But similarly, in an embryological context, you can also have these flows specifically during early development. So these are just two examples where you have flows in a developing chick embryo, and at the bottom, you have a fly embryo. Most often, we think of these sort of flows emerging as a result of chemical cues, which might not be the only reason why they flow. You can also have different mechanical forces that guide these tissue flows. For instance, in the C. elegans oocyte, you could have thermodynamic instability, which actually drives these proteins out of equilibrium and causes them to flow. And in a cell culture context, you could also have different forces that cell-cell and cell-substrate interactions, that is, interactions with the surrounding environment, resulting in these tissue flows. And this could result in a change in matrix property as a result of change in matrix properties or the cells themselves changing their viscoelastic properties as they undergo state transitions. And at the same time, all these flows can also emerge from a possibility of jamming unjamming transitions where as a constraint for space, they start moving and create these out of plane deformations, specifically during gastrulation. So today, what I want to talk to you about is this early stage development of gastrulation, where you have these large scale flows appearing. So, gastrulation is thought to be the most important point in your life, as famously said by Lewis Paul Walpott, because it's the point at which the different germ layers in your body get specified. The germ layers meaning the ectoderm, mesoderm, and the endoderm. But if you look at all the different organisms that are undergoing gastrulation, um, yeah, if you so all of these different organisms, just these are just a couple of different examples highlighted here. They all start from a single cell, and you have multiple cells which, sorry, I'm not sure why my presentation is sort of stuck. Uh, yeah, these single cells undergo an increase in number and are able to form this sort of axis elongation, and you have the different organ structures emerging. But while they go from this single cell or multicellular system into this fully developed organism, they have several processes that they undergo. One being epiboly, which is the process by which they spread over the underlying yolk or the surface that they're growing on. But you also have different modes of gastrulation where they can undergo either ingression or you can have just involution of cells to form the body axis structures. So specifically chick embryos, they start off as a flat sheet of epithelial cells, as shown here. And this flat sheet of epithelial cells have two different regions specifically, which is you have this inside group of cells called as the AP region, which is what gives rise to this primitive streak as you see in this video here, and the different body axis structures. But at the same time, you also have this outer dark region, which is expanding over the yolk and gives rise to this vasculature network over the yolk sac, equivalent structures in the chick embryo. So during my postdoc, I got really interested in this early stage developmental process where you see two different opposing flows. One is this large scale vortex flows in the middle, giving rise to the primitive streak in gastrulation. But at the same time, you have this large scale outward flows or referred to as epiboly. And during this gastrulation process, in this middle region where the primitive streak emerges, you have cells ingressing out to resulting in the formation of the mesoendoderm throughout development. So to understand this process of epiboly in chick embryos, we took a sort of three-prong approach where we started describing the architecture and the mechanical properties that might guide this blastoderm expansion or this epipoly process, then looked at the cellular level processes that drive this expansion, and also to understand how does this expansion then contribute to gastrulation. In the interest of time, I'm only going to delve into the first two bits, but I'm happy to discuss the last aspect as well. So in terms of the architecture, so chick embryos have a unique architecture. You're very well familiar with a chicken egg, where you have this outside hard shell, and you have albumin which surrounds the yolk, and you have the blastoderm which has the fertilized egg sitting on top of the yolk, and you have this glycoprotein membrane called as the Whittlin membrane, which encapsulates the embryo, the yolk from the surrounding albumin. And at the same time, it has a very unique architecture where the circular disc is anchored onto the Whittlin membrane only through the edge, meaning you just have the circular rim of cells which are anchored onto this underlying Whittlin membrane, and everything else is free floating. So we sort of characterize this using electron microscopy. So what you see here is an electron microscopy section where you have just the edge which is anchored onto the underlying Whittlin membrane, and you have free floating cells there. If you look up close, the way it's anchored onto the Whittlin membrane is you have a cell body, you have cell protrusions which emerge on the Whittlin membrane. And at the same time, you have some adhesions which help in anchoring this cell front to the underlying Whittlin membrane. And again, if you go further down into the cell, then you see empty space appearing between the Whittlin membrane and the cell body. What's even more fascinating is the fact that even though it's anchored on the edge here, the cells which are free floating have an apical polarization facing the Whittlin membrane and a basal polarization facing the yolk. So this is a staining of Z01 to illustrate this, where you have the Whittlin membrane on your right and you have Z01 activity or Z01 being polarized onto the side that faces the Whittlin membrane. And when we look at basal lamina similarly, this is a staining for laminin where we see laminin on the basal surface, which is the side facing the yolk in this instance, and on the edge itself is devoid of laminin. So we went on to measure the stiffness of this underlying substrate, which is the Whittlin membrane in this case. And we find that with time, the Whittlin membrane properties change, but it roughly stays within the range of about 50 to 100 kilopascal. So we tried to understand if this expansion or epipoly process itself is guided by the stiffness of the underlying surface. So to do this, we actually modulated the stiffness of the underlying surface by using polyacrylamide gels, removed these embryos out of the Whittlin membrane and grew them on substrates of different stiffness. So when we grow them on soft surfaces, we see an abrogation of expansion, versus when we grow them on stiff surfaces, perhaps due to stiffness sensing of the surface, the cells are able to anchor better and expand better, resulting in an expansion phase. And we did a more quantitative analysis of changing the substrate stiffness, and we do see that the expansion is highly dependent on the stiffness of the underlying surface. So what I've shown you so far is that the expansion itself might be guided by stiffness sensing of the underlying Whittlin membrane. And then we wanted to understand what might be the cellular level processes that drive this expansion. So earlier I showed this data set where we see that the cells on the edge itself do not have any laminin staining. So these are the group of cells that are anchored onto the Whittlin membrane. But when we go further down, we see ECM present in between cells. And if we go further down, we see ECM on the basal surface. So we first went on to characterize the localization of ECM. So these are the top panel, you have the pre-streak stage embryo. And when we look specifically at the edge, you see ECM or laminin in this instance being present as dotted puncta. And once the streak emerges, we see a more fibrous network emerging on the edge. So I wondered if it's the cell movement or the blastoderm expansion guided cell movement which remodels this ECM, because now the ECM is not at the bottom surface, but it indeed is on the top surface. So for this, using antibody injections, I started live imaging ECM in chick embryos. So what you have here in magenta is the membrane label. In cyan, you have laminin. And what you see here is as the cells on the edge or the blastoderm starts expanding, you see ECM movements which follow the movements of the cell. And when we look at the cross-sectional view, we see the ECM which was present mostly as a dotted puncta to be present in between cells, which tends to be localized more onto the basal side as the expansion has undergone. So using particle image velocimetry, what I then did is to measure the cell speed. So on the x-axis here, you have time, and on the y-axis, if I take a single edge, which has a couple of cells in them, and we take an average, and the average is represented as a single line, and we plot this increase in the cell speed over time. This is the kymograph you get. So this is another kymograph for the laminin speed. And when we subtract the two, we see that the speed is almost zero. This highlights that as the cells move forward, they're pulling onto the laminin perhaps and then remodeling them into this sort of fibrous network. So just to quickly summarize what I've shown you today is that this expansion process during blastoderm expansion can be modeled as a collective migration process where you have a unique architecture where the cells on the edge are adhered onto the underlying vitelline membrane through a non-ECM dependent adhesion. And you have a group of non-edge cells which have an apical polarization facing the vitelline membrane. And this expansion itself is guided by, which I didn't have the time to talk about and happy to chat about, is actin mediated process and tubulin mediated process, because when we block these two, we see a reduction in the expansion rate. And this expansion itself is guided by sensing of the stiffness of the vitelline membrane, because when we alter the stiffness, we do see a reduction in expansion. And during this expansion phase itself, what we see is we see a remodeling of ECM, which gets reorganized from this dotted puncta-like structure into a more sheet-like and fibrous network. And as a result, what we think is the reason why the ECM needs to get remodeled is potentially you have cells exiting out of the primitive streak, where it's known that just at the site of primitive streak, you have a breakdown of ECM. But if you do not have this intact ECM, the mesoendodermal cells do not have a surface to migrate over and to create a multi-tissued architecture necessary for further development. And this is evident when we treat the embryos with collagenase on the right, where we see a disruption of the multi-tissue architecture that emerges. So with that, I want to thank everyone in Fungju's lab, specifically Fungju, and Kriti and Venus, who really did a lot of the work to optimize some of the drug conditions and optimize the stiffness measurements, and the Gurdon Institute imaging facility and general facilities for being such a great support over the years, and the EM facility at Cake. And the microscopy facility in Sainsbury Lab, and also specifically the Rosalind Institute in Edinburgh for all the transgenic lines that I've shown you today, and all the funding agencies for their support over the years. And if you have any further questions, I'm happy to take questions now, but also this work is now out as a preprint and it's under revision. So if you are interested in more details, feel free to have a look at it as well. Thank you.
Great. Thank you very much, Lakshmi. We already have a question in the Q&A box, so
Oh,
I'll go
okay.
straight there. So Bruce is asking, did the polyacrylamide gels have adhesive ligands?
Yeah, so in this particular case, we actually added adhesive ligands to it. So we added fibronectin. So we tried a couple of, so Venus, the student who helped me optimize this a bit with her, what we tried is we tried a couple of different conditions. So we tried fibronectin. We also tried just adding albumin, which is what you have in the surrounding environment. And the thing that worked the best was fibronectin. So we also tried a couple of different concentrations, and it seemed to be affected by the concentration of fibronectin to some degree as well.
Great, thank you. And a reminder to everyone to please type your questions into the Q&A box. And while people are doing that, I have a couple of questions to fill the time.
Yeah.
So right at the beginning, you showed that there's two different types of flow in the chick embryo, the sort of the expansion over the yolk and the vortex of the primitive
Yeah.
streak. What happens at the interface between those two flows? Are they physically separated? Is it just cell behavior that determines which way cells go? Can you tell me
Yeah,
about that?
yeah. So I don't think it's really looked into, and this is something I really want to look at to see what happens at the interface a little bit. One thing I can sort of say at the interface, what happens now is it's known that there's a sort of actomyosin cable at the posterior side, which sort of separates where the flows get initiated for this polonase movements and where this outward expansion begins. But beyond that, less is known. From some of the preliminary data I have, I would also say there's some differences in the ECM organization, where it sort of forms a barrier between where these Polonaise movements occur and this outward expansion happens as well. So those are the two sort of clues I have for now. Further than that, I don't really know. And the one thing we also know is we can cut this edge, and you
Mhm.
can still see this emergence of flow appearing. So it seems to be a very robust process. So what you see here on the right is actually an ablated embryo where the edge has been ablated and it's still able to regenerate and um, still sort of recapitulate this complex flows in between. But yeah, I'm not entirely sure up to what depth can I cut it and still see this sort of process. And that's something I'm thinking of doing in the future as well.
Sure, thank you. And you mentioned that the localization of the ECM is really important. Have you also looked at the composition of the ECM and the different ECM proteins that go into that and whether that is important?
Yeah, so I've looked into that, and we do have fibronectin, fibrillin, collagen, and laminin for sure.
Hmm.
I can't be certain about the collagen localization if it matches the laminin localization, but it seemed fibrillin and fibronectin seem to correlate very well with what you see with laminin organization per
Great,
se.
thank you.
Yeah.
So one last question before we move on to the next speaker. Sarthak has asked, well, says they might have missed it, but why was the vitelline membrane removed before the embryo was added to the gel?
Yeah, so the reason we did that was we wanted to see if the cells were sensing the stiffness of the vitelline membrane and to mimic the stiffness of the vitelline membrane, which we knew was about 50 to, sorry, which was between 10 to 100 kilopascal. We tried to mimic that using a polyacrylamide gel so that we can see if we had a lower stiffness, would that then change the properties? And the other rationale that I didn't have the time to get into was also the fact that it's known that the vitelline membrane properties change over time as development progresses. It's known to become softer and more brittle. So, one of the things we wanted to see was if the expansion itself is somehow facilitated by the stiffness changes in the stiffness sensing by the
Great,
cells on the edge.
thank you very much.
Thank
And
you.
with that, we'll move on to our next speaker. Thank you very much, Lakshmi.